Hi, Master John. Hi, Chris. Hi, Linda. And Gabriel. So this is filming this because it's going to be easier than writing a report. In fact, it should be both, but in the end I think I'm just going to give you this video. So I did the first try of the experiments, the microbiology experiments, as I had proposed to Master John in December, and I completed that today. So I will be showing you, we recorded throughout the whole experiments, and then also the results. So I will be showing you what I did in the past 2 days. And we hit a little snag to begin with. We originally I had planned to conduct these experiments at my job's laboratory, but it had scanned that I should ask my supervisor's permission to use the laboratory, and she denied it. So I set up a impromptu micro lab in my home, and that's where we conducted, and I believe we didn't have any trouble with that part. We had to adapt the cupboard under the stairs with a space heater to work as our incubator, and so I called an incubating chamber rather than an incubator. That meant that the bacteria cells were growing at about 10 degrees Celsius below the optimal growth, but it just meant that I just let them grow for longer than usually I would if they were growing at 37 degrees Celsius. So what you'll see next is the experiments and the treatments that I did, and at the end I'll come back and talk about the results. All right, here we go. Okay. So the first thing we're going to do is transfer the liquid Salmonella culture onto the XLD plates. And I'll do that by transferring 100 microliters of the culture. It's in here in this tube, just for ease of handling. "100 microliters is just one big fat drop, but the same volume will be applied to every plate. And with this tool, I spread the bacteria cells evenly across the surface of the agar, so there's no accumulated areas of more or less cells on the plate. This has to be done until all the liquid is absorbed into the agar. Now I'm sterilizing this tool between applications, between uses, and it sits there to cool down. So this plate is ready. Now it contains the Salmonella cells, and I will repeat this process for all 30 plates that are required to perform this experiment. So I'll begin by making a invocation, invoking a blessing for this experiment in honor of the sacredness of this moment. Supreme God, Masters and Angels, Master John Douglas, I lovingly request your blessings for this experiment. May it reveal truth in matter. May this empiric demonstration of Supreme God's power support Master John Douglas' mission on earth and be of benefit to all. Thank you, Masters, thank you, in gratitude and full faith. Okay, we're going to begin by testing the food repair disc that I have here. And we'll begin with 5 seconds exposure in the 3 plates that are marked 5 seconds. And I will begin a timer. 5 seconds. So I'm going to hold the food repair disc under the glass for 5 seconds. That's that. The next exposure is 10 seconds. The next one is 15 seconds. And the last exposure of the food repair disc is 20 seconds. That's 10. That's 20. So these are the 4 treatment doses for the food repair disc that we are testing. Cut. Alright, the next part of the experiment is the killing requests to the Master Angels. The first one is the 25% or lethal dose 25. I will make the request now. Masters and Angels, I humbly request that you kill 25% of the Salmonella bacteria in these 3 plates marked LD25. Thank you, Masters. Thank you with gratitude and full faith. The next one is lethal dose 50. Masters and Angels, I humbly request that you kill only half or 50% of the Salmonella bacteria in the 3 plates marked LD50. Thank you. Thank you with gratitude and full faith. Next one is LD100. Masters and Angels, I humbly request that you kill all 100% of the Salmonella bacteria present on the 3 plates marked LD101. 100. Thank you, Masters. Thank you. Thank you with gratitude and full faith. The last dose will be actually, the last treatment is actually no dose requested. Masters and angels, I humbly request that you kill the Salmonella bacteria on the 3 plates marked no dose. Thank you. Thank you with gratitude and full faith. And these were the 4 treatments for the killing request. And we have 3 plates of control that received no no treatment, no killing requests, or no food repair disc treatment. And we have another 3 plates of controls, and these actually contain only half of the amount of bacteria originally placed in all plates. In case there is too much growth in the control, we'll be able to count the number of colonies more easily if it's a half dilution. So this is also a control. We have a total of 6 control plates. And that is the end of the treatment. Now every plate will be placed in the incubating chamber, and they will be allowed to grow overnight, and we'll see the results tomorrow. Okay, so the plates were incubated for 23 hours at 26 degrees. And what we're looking here are the 6 plates that were our control plates. On the top row we have the diluted initial 100 microliters of culture diluted in 100 microliters of PBS, just to cut down the amount of cells by half. And here we have the plates that have the exact same amount of cells as our tested or treated plates. So what we see here is what I was trying to avoid, is what we call a lawn. So we don't see individual colonies. We see as the entire surface of the agar is covered by cells. And you see the difference in colors in the red and the yellow is based on the metabolism of the bacteria that is breaking down the nutrients in the agar and changing the pH of the medium, and that causes the change of color. In this row here we can actually see the black is the telltale sign of Salmonella. Salmonella also on this type of media will produce these black colonies. So you can see that there is a lot of bacteria here, and I will not be able to count individual colonies. That will then be useful for doing statistical analysis. So this was an unfortunate result. And now we're going to look at the plates from the experimental treatments. These 6 plates were treated with the food repair disc. Can you come closer? Yes, thank you. And what we see is the same problem that we had on the control plates, that the bacteria formed a lawn on top of the surface, and we don't have individual colonies. These are the plates that were treated for 5 seconds. The same thing we see in the plates that were treated for 10 seconds. They're all covered in Salmonella, and we are not going to be able to count the number of colonies. And it turns out that this happened for all plates. We have lawns throughout. This is 15 seconds treatment on the plate, on the disc. These are 20 seconds, and they are just completely covered with bacterial cells. The plates that contain the cells that were treated with killing requests, it's the same type of situation. Can you close? They're lawns. So all in all, this experiment did not work, and I would attribute that to a very large initial inoculum of the bacteria on the plate. So I should probably have used maybe 1/10 or even 100th of the of what I placed here, so that we would have single colonies, which means to acquire a single colony you have to have one individual cell that is alone in an area of the agar, and that cell will then multiply over the night and form a visible individual colony. So what we have here is too many cells, and we are unable to count. And also probably it was too many cells even for the treatments that we were performing. So this experiment will have to be repeated, and we will have to reevaluate the experimental design to move forward. Okay, so now you've seen that the experiment didn't work, and that was a technical fault, and it was my fault, I believe. I have not scanned for that yet, but I believe that I should have begun with a much smaller number of cells that were plated on the agar, and I should have diluted the initial liquid culture pretty far before putting it on the agar plates, and that would give us a small enough number of cells that would allow for single colonies, because as I explained, we need one cell, individual cells, to be spread out the plate, and then each cell will multiply and form a colony the next day, and so we can count. When there's just too many side by side, they will start multiplying, and then they will spread out and connect to the colonies next to them, forming the lawn as we observed. So my plan is to get new plates. I still have the bacteria. We'll get more plates, and I will begin with a smaller, simpler experiment this time. I've done this in the past, of trying to begin with a more complex experiment to begin with. So I'm gonna take a step back and test only the food repair disc, and I will try different dilutions of the bacteria with the different treatments. So hopefully one of the dilution factors will give us the individual colonies that we are looking for. So until next time.